Good afternoon. My name is Vahid Graywal. Over the summer, I spent 8 weeks studying liver cancer using the zebrafish model in the Sadler lab at Mount Sinai. Liver cancer is the third leading cause of cancer-related mortality worldwide, but unfortunately, there's no cure for it. Last year, I did a project on organ donation rates, which remain extremely low in our country. My research made me realize how vital it is for us to develop alternative treatments for liver cancer so we're not as dependent on organ donors for liver transplants. The gene I focused on was UHRF1. UHRF1 is a protein-coding gene and an epigenetic regulator that is involved in DNA methylation and histone modification. It is also required for liver growth and regeneration. However, UHRF1 is highly expressed in patients with advanced liver cancer. While previous researchers in my lab have confirmed that UHRF1 is in fact a driver of liver cancer, the exact mechanism behind its oncogene function is still very unclear. So to further investigate this, in my study I analyzed transgenic zebrafish livers to elucidate how overexpression of UHRF1 correlates with hepatocyte cell division and contributes to the development of liver cancer. So just to start off with a few basic definitions, epigenetics is the study of heritable changes in gene expression that are not caused by actual alterations to the DNA sequence. One important epigenetic system is DNA methylation. DNA methylation is the addition of a methyl group onto the cytosine base. During DNA replication, UHRF1 recognizes hemimethylated DNA and recruits an enzyme called DNA methyltransferase to maintain methylation in daughter cells. A transgenic organism has a gene from another organism artificially inserted into its genome. Zebrafish happen to be ideal model organisms because not only are they vertebrates, but they're estimated to be 60 to 75% genetically homologous to humans, where they have very large progeny. Some of the fish I was working with were laying 200 to 300 eggs at a time, and their embryos are transparent, which makes them very easy to study and genetically manipulate. I work with transgenic zebrafish that overexpress human UHRF1. The human UHRF1 is tagged to a green fluorescent protein abbreviated as GFP so that it's easy to locate and assess. The human UHRF1 was under the hepatocyte-specific promoter FAPP10. For the purpose of my study, I refer to these transgenic fish that overexpress human UHRF1 as UHRF1 high, and in many of my analyses, I compare them to wild-type zebrafish livers. So my first hypothesis was that in UHRF1 high zebrafish livers, the level of UHRF1 overexpression would increase between 3 and 10 days past fertilization due to increased activity from the FAPP10 promoter. Along with this increase in expression, I hypothesized that the nuclear morphology and cell division would be affected in the UHRF1 high livers. So in order to assess how UHRF1 overexpression affects cell division, I treated 3, 4, and 5 days past fertilization with wild-type and UHRF1 high zebrafish embryos with BRDU. BRDU is a nucleoside used to tag cells that are dividing. I then anesthetized them and fixed them. My mentor provided 10-day-old both wild-type and UHRF1 high zebrafish embryos that were previously incorporated for BRDU. I then conducted immunofluorescence on all the fixed embryos in order to analyze the BRDU nucleoside. Then I dissected the livers out of the UHRF1 high zebrafish embryos and mounted them onto slides. I imaged all my liver samples using a Leica confocal microscope. I then used a program called ImageJ to analyze my liver images. For each time point, I measured about 10 wild-type and 10 UHRF1 high livers, and within each liver, I analyzed approximately 100 to 200 hepatocyte nuclei. The conditions I measured for were UHRF1 overexpression via the green fluorescent protein fluorescence, DNA staining intensity via the Hox DNA stain, circularity, nuclear area, and cell division via the BRDU fluorescence. So in this first panel here, I have the wild-type livers on the left and the UHRF1 high livers on the right. The time points are 3, 4, 5, and 10 days past fertilization. In this first figure here, I compared the nuclear area between wild-type and the UHRF1 high livers. On the y-axis, I have the nuclear area in micrometers. As you can see in this box and whisker plot, the nuclear area was significantly higher in the UHRF1 high livers at all 4 time points. I hypothesized that perhaps the nuclei were larger in the UHRF1 high livers because there was more DNA content. I did some further reading on the Hox DNA stain, and I learned that it actually intercalates into the DNA because it binds to the minor group of the DNA strand. I then decided that since I had the nuclear area of each hepatocyte nucleus, and I had the average Hox fluorescence, I could multiply these two values to approximate the total DNA content within a hepatocyte nuclei. I could then compare the relative DNA content between the wild-type and the UHRF1 high zebrafish livers. So as you can see here in this box and whisker plot, the relative DNA content was actually significantly higher in the UHRF1 high livers at 3, 5, and 10 days past fertilization, which bolsters my hypothesis. I also read that the Hox DNA stain is generally bright when the chromatin is tightly packed and dim when chromatin is loosely packed. Interestingly enough, I saw that the Hox fluorescent was actually dimmer in the UHR1 high livers at all 4 time points. This is suggestive of euchromatin, or loose chromatin structure, which is counterintuitive because since there is more UHR1, you would expect that there would be actually heterochromin or tighter DNA, but this is actually the opposite, as you can see here. In this last box in this plot, I compared the circularity on a scale from 0 to 1, with 1 being a perfect circle. I found that at 4, 5, and 10 days past fertilization, the hepatocyte nuclei were significantly less circular in the UHR1 high livers. Especially if you see here, at 10 days past fertilization, the nuclei are very abnormally shaped and disfigured, and they barely resemble their wild-type cohort. Abnormal morphology is a hallmark of cancer, which leads me to believe that perhaps at 10 days past fertilization, tumorigenesis is beginning to set in the UHR1 high livers. In the second panel here, I have the wild-type livers on the top and the UHR1 high livers on the bottom. The blue represents the Hox DNA stain. The green in the UHR1 high livers is the green fluorescent protein of the UHR1, and the red is the BRD cell division stain. I compare the mean percentage of dividing hepatocytes between the wild-type and UHR1 high livers. As you can see here, there is no significant difference in cell division at 3 days past fertilization, perhaps because this is the time when the gene is first turned on. However, at 4 days, while there was approximately 17% cell division in wild-type livers, there was only 1.3% in the UHR1 high livers. Further, by 5 days, while there was still 12% cell division in the wild-type livers, there was almost no cell division in the UHR1 high livers. However, at 10 days, while cell division had ceased in the wild-type livers, it was certainly beginning to pick up again in the UHR1 high livers. After seeing these anomalies in cell division, I did some further analysis on solely the UHR1 high livers. Here I have 4 UHR1 high livers at 3, 4, 5, and 10 days past fertilization. I compared the mean level of UHR1 expression over the 4 time points. I found that the level of UHR1 gradually increases and peaks at 5 days past fertilization, which is shown by how much brighter these nuclei are compared to the other 3 time points. The level of UHR1 begins to then decrease by 10 days past fertilization, as these nuclei are clearly dimmer than these nuclei. I then looked at an individual 4-day UHR1 high liver and an individual 10-day UHR1 high liver. I divided the hepatocyte nuclei based on whether they were positive or negative for the BRD cell division marker. I found that those cells that were negative for the UHR1 high, were negative for the cell division, had a very high level of UHR1. On the other hand, while those that were positive for cell division had a very low level of UHR1. This led me to believe that the level of UHR1 overexpression is inversely correlated with whether or not a cell divides. I summarized my findings in terms of cell division and UHR1 expression into these 2 graphs. As you can see, the level of UHR1 overexpression gradually increases and peaks at 5 days and then decreases. On the other hand, the level of cell division did the exact opposite. Cell division decreases abruptly up to 5 days and then begins to start up again at 10 days. So my current working model is that UHR1 overexpression leads to senescence at 5 days past fertilization. Senescence is when a cell loses the ability to divide. This senescence may be related to the hypomethylation that I was seeing here in this first panel. Then I believe that tumorigenesis occurs at 10 days past fertilization, bolstered not only by the increase in cell division, but also by the abnormal morphology. This summer, I will be returning to the lab to test additional time points between 5 and 10 days past fertilization to determine how cells are either escaping senescence or whether some cells are not actually senescing and they're becoming cancer cells right away. I will also be testing time points after 10 days past fertilization to see if and how the tumors actually develop. Additionally, I will be learning how to conduct flow cytometry so I can more accurately measure the DNA content and gauge which phase of the cell cycle all the cells are actually in. Thank you so much for listening, and be happy to answer any questions that you have.